everybody, welcome to the muscular system. This is part 1, and before we get into the muscular system, I want to begin with this overview integration slide. Alright, so we started the semester by looking at what is in a single cell, right? And we see that the main component of a cell is water, and that led to the discussion of hydrophilic and hydrophobic interactions and amphipathic interactions. And so then we looked at the cell membrane, and we see that the cell membrane is made up of amphipathic molecules, namely the phospholipids. And in the cell membrane you have lots of proteins. And then the question was, where do proteins come from? And we talked about the central dogma, and then we saw how proteins are processed and transported as well. And then we talked about how individual cells are linked together by cell junctions, and we talked about 3 types of cell junctions, and we will be talking about at least 2 of these cell junctions in the muscular system. Then we looked at the various types of tissues. We looked at the epithelial and the connective tissue, and we'll be revisiting how connective tissue is found in the muscular system in this set of lectures. After that, we moved on to the integumentary system, where we saw all 4 types of tissues. The muscular, nervous, epithelial and connective tissue all are found in the integumentary system. And then we moved on to the skeletal system, and we see that the way to improve bone density is by engaging in weight bearing exercises, and to perform weight bearing exercises you need to use the muscular system. And then we looked at the endocrine system, which explains how the various parts of the body communicate with each other through hormones being sent into the bloodstream. And we also saw that the nervous system is another way in which the various parts of the body communicate with each other through signals sent through the nerves. And then yesterday we looked at the differences between the sympathetic and the parasympathetic division of the autonomic nervous system. And this parenthesis here is indicating that pretty much all of the organ systems which we talk about from this point onwards will be controlled by the nervous system, right? And we will look at how signals sent from the central nervous system will be helping the muscles to contract, right? And how, once a sensation is detected, how does the body respond? We'll better understand that by looking at the functioning of the muscular system. So with that, I'd like to begin with an overview of what I hope to cover today. So we're going to talk about the 3 types of muscle, the cardiac, skeletal and the smooth muscles, but the main focus in this muscular system will be on the skeletal muscle. And I will point out the structural organization of the skeletal muscle. We look at starting from the big picture, all the way from the organ level down to the organelles in the cells which help the functioning of the muscle cells, the entire muscle. And then we look at what is known as cross-bridge cycle, which will take place inside the muscle fiber or muscle cell or myocyte. Those names are all interchangeable. We'll talk about what is the cross-bridge cycle. And then we're going to again zoom back out and look at the excitation-contraction coupling. So excitation is the signal being sent from the central nervous system to the muscle, and how that is going to be coupled with the contraction of the muscle in response to signals from the brain. After that, we'll look at some of the differences between the cardiac and we'll look at some features about the cardiac and the smooth muscles, and we'll see how there are some structural and functional differences when compared to the skeletal muscle. So with that, let's talk about how muscles are classified. So muscles can be classified into 2 broad categories based on their appearance. You have smooth muscles and striated muscles. And striated muscles have this stripe-like pattern, or they have these striations. And among the striated muscles, you can divide them into skeletal and cardiac muscles. And the reason for these striations is the presence of the structural and functional unit, which is known as the sarcomere, right? So sarcomere is a structural and functional unit in the muscle, and you have those sarcomeres in skeletal and cardiac muscles, but you don't have that in the smooth muscle. And so there are some similarities between the cardiac and skeletal muscle, but there are also some major differences. For example, you have no control over the contraction and relaxation of the cardiac muscle, but you can control many of your skeletal muscles. So with that, let's take a closer look at the structure of the skeletal muscle. So we see that the skeletal muscle is going to be connected to the bone by means of tendons, right? So do you remember what type of connective tissue makes up tendons? Dense, regular connective tissue. Yes, thank you. Can't forget what we learned in Unit 1, all right? So dense regular connective tissue will connect the muscle to the bone. And if you take a look at the muscle, we see that it's made up of these various bundles known as fascicles. Fascicles or bundles can be interchangeably used. And then if you were to tease apart what is making up these bundles or fascicles, we see that it's made up of individual myofibers. And as I mentioned, the term myofiber can be interchangeably used with myocyte or muscle cell, right? It's the same thing. So when you take a cross-sectional view of the entire muscle, I want to point out that there are some layers holding these structures together. So the outermost layer is known as the epimysium, and that is what is going to surround the entire muscle. And I told you that the entire muscle is made up of a number of bundles, and those bundles are surrounded by connective tissue, which is known as perimysium. And then each of the individual myofibers or muscle fibers, they're going to be surrounded by the endomysium. And all of these layers, the endomysium, the perimysium, and the epimysium, they're all connective tissue, right? And they'll eventually join together to form this tendon. Okay, so you also notice one of the key features in the skeletal muscle is that it is multinucleated, right? It's got lots and lots of nuclei. And the question is why? The reason is the way these muscle cells are formed is by the fusion of these individual cells known as myoblasts. So multiple myoblasts, they're going to come together and fuse, and that's how it leads to the formation of the entire muscle cell. And the interesting thing about the muscle cell is that it is very long. It's going to extend for the entire length of the muscle. So it can be up to 30 centimeters in length, right? So you can just pull out an entire muscle cell and it looks like a fiber. So now let's take a closer look at the structure of an individual muscle cell. We know the length is 30 centimeters long, up to 30 centimeters long, depending on where in the body it's been extracted from. But let me point out something in regards to the dimensions of the diameter. So the diameter is only going to be 100 micrometers, right? So it's very thin. And the outermost layer of this muscle fiber is known as sarcolemma, and cytoplasm, which is inside the muscle fiber, is known as sarcoplasm. Right, so the word sarco is going to be used over and over again because it just means muscle or flesh. Now we see that this sarcolemma has got invaginations into the sarcoplasm in these structures known as T-tubules. So they look like little tunnels passing right through the muscle fiber, and these are known as T-tubules. And we know that the analogy I would give you is that these T-tubules are like tunnels going through mountains, right? And so when you think about a mountain with tunnels, what is the first thing which comes to your mind? What goes through those tunnels? What? Cars, trains, right? So something's being transported through those tunnels. And so what is going to be transported through these T-tubules will be action potentials, right, which will be generated as a result of signals from the brain. Now, right adjacent to these T-tubules, you have these large sac-like structures known as sarcoplasmic reticulum, and this is a modified type of smooth endoplasmic reticulum. And what the sarcoplasmic reticulum contains is lots and lots of calcium. Right? We talked about smooth endoplasmic reticulum for maybe 30 seconds in Unit 1, but I told you it will help store calcium. and here's where we'll be talking about it. Now we see that in between the T tubules and the sarcoplasmic reticulum, you have two types of receptors, right? You have these dihydropyridine receptors, which are towards the T tubules, and towards the sarcoplasmic reticulum, you have the ryanodine receptors. And the interesting thing about the dihydropyridine receptors is that they are voltage gated. They are sensitive to changes in voltage, and so as the action potential is going to travel down the T tubule, it will activate the dihydropyridine receptors, and the dihydropyridine receptors will then interact with the ryanodine receptors to cause calcium to be released from the sarcoplasmic reticulum, right? So that's how these two receptors work together to regulate calcium release from the sarcoplasmic reticulum when an action potential travels down the T tubule. Okay, and then we'll see that each of these T tubules, they're going to surround these structures known as myofibrils, right? So that is another important thing to consider. You have these many myofibrils which are surrounded by these T tubules. Now when you take a closer look at the myofibril, we see that it is made up of these repeating units known as the sarcomere, and the sarcomere is the structural and functional unit of the muscle. Whatever happens at the sarcomere is going to happen happening throughout the muscle to cause it to contract. Now notice that this sarcomere is only like 1 to 2 micrometers in diameter, and we see that there are two key features in the sarcomere which I want to point out. You have the thick filaments shown here in purple. They look thick, and as the name indicates, you have these thin filaments shown here in red, right? And so the thing to note here is that there is a region of overlap between the thick and the thin filaments, right? So there's a certain region of overlap between the thick and the thin filaments, and it is this overlapping regions of the thick and the thin filament is what leads to the striated appearance in the skeletal and cardiac muscle. So I told you that the reason for the striations is due to the presence of sarcomeres. Sarcomeres have thick filaments and thin filaments, and they're going to overlap with each other, and it's that region of overlap which leads to this striated appearance. So now we're going to take a closer look at the sarcomere, and I want to point out a couple of things. Right. So we see that on the top is a cartoon of the sarcomere, and below is the electron microscopic image of the sarcomere. And notice that this region of overlap, the dark region, is called as the A band, whereas you have this region where you have only thin filaments, right? There's no thick filaments. You have only thin filaments. That's called as the I band. So the A band is the dark band where you have thick and thin filaments overlapping. The I band is where you have only thin filaments. A couple more things I want to point out to you is that this dark line in the middle of the I band, it's known as the Z line. So the Z line is made up of various types of proteins, and it's the Z line to Z line which is making up a single sarcomere. So the thin filaments are all connected to the Z line, and from Z line to Z line is a single sarcomere. Now in the middle of the A band, which is the dark band, you find there is a M line, which M stands for middle line, and at the M line you have all of the thick filaments being connected one to another, right? And so these are the features I wanted to point out, and it's actually possible for the thick and the thin filaments to slide across one another as the muscle contracts, and we'll see how that takes place. Any questions up to this point? We're just looking at the structure of the sarcomere. We worked all the way down from the muscle as an organ down to what is making up the various components of the cell. Yes?
So just to clarify, the H band is where the thin and thick filaments are able to connect with one another, while the M line is just the thick filaments connected together.
Yeah, so the question about the H band. So the H band is, it's where you don't have any thin filaments present. Yeah. So the thin filaments are not found in the H band, as you can see here. It's only the thick filament. But the M line is the region where all the thick filaments are connecting one to another. Right. So now let's move on. Okay, and by the way, if you are curious about why one is called A band versus the other one called I band, it's beyond the scope of our discussion. It's how polarized light passes through this region, right? So it's something beyond the scope of our discussion. Anyway, so now let's take a closer look at the structure of the thick filaments. We see that the thick filaments, they are made up of these myosin molecules, right? And these myosin molecules, they have two heads. So, and this tail is where you have the individual myosins wrapped with each other. So a single myosin has got two heads and it's got this long tail. And notice that the tail is oriented towards the M line, whereas the heads, they are oriented towards the Z line, right? So the tail is towards the M line, the head is towards the Z line. And each of these heads, they have two things I want to point out. They have a site where ATP can be bound and hydrolyzed, and that will energize the myosin head, and we'll see how this plays a role in muscle contraction later on. But secondly, there's also a site where it can bind to actin. Right? You remember we talked about actin when we discussed about cytoskeletal proteins. We also see that these actin will play a role in muscle contraction. So the thin filament is made up of these actin subunits, right? And the way the actin subunits are arranged on the thin filament is that they are arranged in a helical structure. So it's like a string of beads wrapped around each other. That's how these actin subunits are arranged in the thin filament. Now, what controls the myosin, whether or not it binds to the actin subunits, is the presence of the regulatory complex. So there is a protein complex which will determine whether or not the myosin can bind with actin. And so this here is the long tropomyosin protein, and the tropomyosin is a long slender protein, and it's going to span for 7 actin monomers. That's how long it is. And you have these tropomyosin proteins which are arranged head to tail, right? They are arranged head to tail, and there's a slight overlap between the tropomyosins. And the place where there is this head to tail overlap of the tropomyosin molecules is where you have another protein complex known as the troponin complex. And this troponin complex has got 3 subunits, right? So it's got the TNC subunit or troponin C subunit shown here in red. It's called TNC because it binds calcium. Then you have the TNI subunit, which has an inhibitory effect, and it will inhibit interaction between this myosin and actin when there is no calcium. And there's also this TNT or troponin T subunit, which tethers TNC and TNI to the tropomyosin protein, right? So because it's tethering the other 2 subunits to tropomyosin, it's called troponin T. Okay, so we see that there will be structural changes taking place in this troponin-tropomyosin complex, which leads to whether or not the myosin interacts with actin. So we see that calcium is going to trigger changes to the troponin complex. So here's the troponin complex shown here. This is another representation of the troponin complex, right? And these changes eventually allow actin to bind to myosin. So here is the troponin C subunit shown here in red, and I just want to point you that there are different regions, right? I'm not going to ask you to memorize this, but just notice that when calcium binds to the TNC subunit, here you have calcium shown here as yellow circles, they bind to troponin C subunit. Notice that there is this hydrophobic pocket in the TNC which opens up. And the opening up of this hydrophobic pocket causes the TNI, which has the inhibitory effect. It's going to now no longer bind to actin, but it binds to troponin C. And this structural change will lead to other changes which permit actin and myosin to interact. All right, so let's see how this takes place. So what you're seeing here is an end-on view of a single actin filament, right? So The actin filament is shown here in yellowish green, right? And then you have one troponin complex here, and then this tropomyosin is shown here. So you're standing at one end here and you're looking all the way down, and if you can see all the way in the distance, you have a tiny troponin complex. So you have these troponin complexes all throughout. And what you see here is a myosin head shown here in purple, and this myosin head is also known as a cross bridge because it's forming a bridge between the thick filament and the thin filament, because this myosin head can bind to actin. And we see that in this current state, this tropomyosin is blocking myosin from interacting with actin. But then when calcium is present, we see that calcium will bind to the TNC subunit. TNI is then going to bind to TNC, and then this tropomyosin will slightly move on the actin filament, thereby causing this myosin head to weakly bind. But this weak binding of myosin to actin is not sufficient to produce force, right? And so as we have more and more of these myosin cross bridges weakly binding, that will cause this tropomyosin to move further along the actin filament, and thereby the myosin can bind strongly, and the strong binding of myosin to actin is what leads to force production. So these three states of the thin filament, meaning the three states at which this troponin tropomyosin complex exist on the actin filament, we can consider it as the three functional states, right? So the first state is the blocked state, where myosin cannot interact with actin because tropomyosin is blocking it. And then you have this closed state where you have a weak interaction between actin and myosin thanks to the structural changes made by calcium. And then you have this open state where the cross bridges will push the tropomyosin further, and then actin and myosin can strongly bind, and that leads to force production, right? So you have this simplified three-state model to explain how there are changes made to the thin filament due to calcium and the myosin cross bridges as well. Okay, any questions? Okay, so here is a polar viewer question for you, right? So we see that a mutation in troponin C is going to decrease its capacity to bind to calcium, right? So in such a case, which state of the thin filament is going to be promoted by this mutated TNC? Would it be the block state, closed state, or the open state? You can ask your neighbors or turn to your TAs and we'll talk about this. Okay, Harrison, do you have anyone in your group who wants to take a stab at it? Oh yes, right here.
We said blocked.
Okay, all right. Most of you said block state, and that is correct, right? So let me explain why this is the case. So we see that calcium is needed to move the thin filament from the blocked state to closed state, right? And if calcium is not able to bind to troponin C, then we see that it's going to, the thin filament will remain in the block state. So that mutation is going to promote the thin filament being in the block state. All right. So now let's watch a quick video on what is meant by the cross-bridge cycle, because just like how we had changes in the thin filament, even in the thick filament there will be changes taking place as well. And this short video, it's a very simplified version of what we'll be discussing. So we'll watch what happens and see how the muscle contracts, and then I'll give you some additional details as well. All right.
Of repetitive functional units called.
Okay, let's play that again.
Skeletal muscles are composed of muscle fibers, which in turn are made of repetitive functional units called sarcomeres. Each sarcomere contains many parallel overlapping thin, actin, and thick myosin filaments. The muscle contracts when these filaments slide past each other, resulting in a shortening of the sarcomere and thus the muscle. This is known as the sliding filament theory. Cross-bridge cycling forms the molecular basis for this sliding movement. Muscle contraction is initiated when muscle fibers are stimulated by a nerve impulse and calcium ions are released. The troponin units on the actin myofilaments are bound by calcium ions. The binding displaces tropomyosin along the myofilaments, which in turn exposes the myosin binding sites. At this stage, the head of each myosin unit is bound to an ADP and a phosphate molecule remaining from the previous muscular contraction. The myosin heads release these phosphates and bind to the actin myofilaments via the newly exposed myosin binding sites. The two myofilaments glide past one another, propelled by a head-first movement of the myosin units, powered by the chemical energy stored in their heads. As the units move, they release the ADP molecules bound to their heads. The gliding motion is halted when ATP molecules bind to the myosin heads, thus severing the bonds between myosin and actin.
Okay. So those are just some very simplified explanation of the events, right? What we just now saw. But there is something not accurate in that video, and that video just did not talk about the myosin head binding weakly versus binding strongly. It just went from unbound to bound. And I want to explain the cross-bridge cycle in a 3-state model of the myosin heads as well. So we're going to see what happens when the myosin head is detached, when it is weakly bound to actin, and when it is strongly bound. So we see that in the detached state, the myosin head has bound ATP, and that ATP has been hydrolyzed to ADP and inorganic phosphate. And we see that this detached cross bridge is going to align with the blocked state of the thin filament, right? Notice that tropomyosin is blocking interaction between actin and myosin. But then when calcium is present, calcium binds to the troponin C subunit of the troponin complex, causes structural changes, and thereby allows tropomyosin to slightly move on the thin filament, thereby allowing the myosin head to weakly bind. And in its weakly bound state, notice that the inorganic phosphate is still present in the myosin head. But then it's the release of this inorganic phosphate which will permit the myosin head to transition from this weakly bound state to the strongly bound state. And notice that this myosin head is ratcheted forward, and it is pulling the thin filament towards the center of the sarcomere. So it's pulling the thin filament towards the center of the sarcomere, towards the M line. And this myosin head is going to remain bound to the actin filament until a new ATP molecule comes in here, kicks out the old ADP, and then the myosin head has now been re-energized, and it can be ready to engage in another cycle of interacting with actin. Right, so we see that this 3 states of the cross bridge, they will align with the 3 states of the thin filament, right? And this is how the contraction will eventually take place. As these myosins interact with actin, they are going to cause the thin filament to slide over the thick filament, and that leads to contraction of the sarcomere. So if you were asked to make a solution, right, to make the muscle contract, which would you add in here? Would you add ADP, inorganic phosphate? Would you add maybe EGTA, which will remove calcium? Would you add magnesium, calcium? What do you think is necessary for the muscle to contract? Since you're running short of time, can someone yell the answer? What would you add? What? Was it? Calcium. Yes, calcium is the right answer, right? So adding calcium is necessary for the muscle to contract. So now, if you want to make the muscle relax, right, we can get rid of the calcium by using EGTA, right? So it's a calcium chelator which can bind calcium, and so that we can get rid of the calcium. But we need another very important component for making the muscle relax. And what do you think that component would be? Would you need ADP, ATP, magnesium, calcium? You should be able to answer this question based on the CrossFit cycle. Anybody? It's ATP is the right answer, right? And the reason why we need ATP is because ATP, when it binds to the myosin head, that's when the myosin head will detach from the actin filament, right? That's why ATP is a very important factor for muscle relaxation. And then if you can answer this question, then I know that you have understood the CrossFit cycle. So now let's talk about the excitation-contraction coupling, right? And so this here is something which you should all be very familiar with, since we just talked about the nervous system. What you see here is a neuromuscular junction, and this membrane here is the sarcolemma. This is the membrane surrounding the muscle fiber, and this is the presynaptic terminal of an efferent motor neuron. So efferent motor neurons carry signals from the central nervous system to the muscle, causing it to contract. And once this presynaptic terminal receives the action potential, it's going to cause the release of acetylcholine from the vesicles. And how are the vesicles releasing acetylcholine? It's through the synaptic complex. And acetylcholine will bind to ligand-gated ion channels shown here in this neuromuscular junction, which will cause sodium ions to enter in, right? And the entry of sodium ions from the opening of these ligand-gated ion channels will cause the threshold to be reached, right, around -60 to -55 millivolts. And we see that this will cause the opening of the voltage-gated sodium channels, leading to a rapid depolarization of the membrane potential in the muscle cells. And we see that around +30 millivolts, even in the muscle cell, you have the closing of the sodium channels by the inactivation gate, right? So the inactivation gate closes, but the voltage-gated potassium channels will open up, and that leads to a rapid efflux of potassium ions from the cell, leading to the repolarization. And notice that because the potassium channels don't close as quickly, it can lead to hyperpolarization as well. And we'll just talk about how the muscle is going to contract now, right? So as the action potential travels down the T tubule, we see that the dihydropyridine receptors, they are sensitive to changes in the voltage. They will interact with the ryanodine receptors and cause calcium to be released from the sarcoplasmic reticulum. The calcium will bind to the troponin complex in the thin filament, permitting interaction between actin and myosin, and that leads to the muscle contraction. So when we come back tomorrow, we will talk about how relaxation takes place. Thank you all for your time. Do you have a question?